Hello friends, very proud and recently the biggest miracle of medical science has happened. UK researchers have created such a super baby, who has the DNA of not 2 but 3 different people. Meaning he was born not to 2 parents but to 3 parents. This means that his superpower is strong immunity. Because now, due to this technique, it is being claimed that he can never become victim of any mitochondrial genetic diseases. Yes, apart from normal genetic diseases, mitochondrial genetic diseases are also a grave problem. Look, you might not know this, but the mitochondria present in every cell, as you might have heard, is the powerhouse of our cells because ATPs are produced there. ATPs means adenosine triphosphate molecules which provide us energy.
Mitochondria also has its own different DNA,
which is different from the nuclear DNA, i.e., the DNA present in every cell nucleus. And a study revealed that if there is any mutation in this DNA, or the mother's defective mitochondria are transferred to the baby, then those babies become victims of mitochondrial diseases. Now here I have specifically talked about the mother's DNA because the mitochondria of the sperm are present here at the beginning of its tail. So during fertilization, when the sperm and egg cells fuse, then all the parts, except the head, get degenerated. So ultimately only the mother's mitochondria remain inside the baby. To investigate the cause of mitochondrial defects, researchers analyzed the egg cells from 21 women aged 26 to 42. In this study they found that only one female egg cell in the age group of 26 to 36 had mitochondrial mutations, whereas in the age group of 37 to 42, 6 females had mitochondrial mutations in their egg cells, and this led to the conclusion that with increasing age the chances of mitochondrial defects also increase, and if these females gave birth to children at this age, then the probabilities of their children having mitochondrial disorders increase. And this is because our human body works like a machine, and like all machines, in this too the error rates keep increasing with time. For example, take the core process of our body, DNA replication. In this, when 1 million nuclear bases are arranged, an error of 1 base or simply a wrong base is made, and the rate of these errors keeps on increasing with age. This occurs in mitochondrial DNA as well. The only difference here is that the error rate here is already 1,000 times higher than that of nuclear DNA. In fact, nuclear DNA is protected by a lot of enzymes and proteins. Mitochondrial DNA does not have this protection, and because of this, mitochondrial DNA gets damaged more quickly, leading to many fatal diseases. Take the case of Jessica for example. So Jessica has a mitochondrial disease, Leig's disease, due to which her brain has started rotting, and because of this her motion development has slowed down, and today unfortunately she has to be fed through tubes. According to doctors, there is no such treatment available in the medical field that can completely improve Jessica's condition. In fact, he even says that she will not be able to survive for long, because mitochondrial diseases affect the cells of any vital organ of the body like brain, lungs, heart, kidneys, and damage them in such a way that people develop life-threatening diseases like cardio diseases, diabetes, liver and kidney failure, ultimately which becomes the cause of his death. Now out of all these diseases, cardio diseases in particular caused the most deaths. As you can see in this report, in the UK alone, more than 150 children are dying every year due to mitochondrial mutation. And if I talk about the overall world, then 1 in every 5,000 dies due to mitochondrial diseases. But friends, all this hopefully no more. Because scientists have found a great way to remove such irreversible damages. The same method which I introduced to you earlier in this video.
3-parent baby. To create these 3-parent babies, scientists first studied the mitochondrial process in detail. He observed that in order to make the enzymes required to make ATP in mitochondria, they need to be produced quickly. So cells store their information not in the DNA of the nucleus, but in the DNA present inside the mitochondria, and if these enzymes are not produced on the spot, the body stops functioning, like what happened in Ben's case. In fact, 11-year-old Ben had a fatal genetic disorder called hypotonia, in which the muscles become soft, due to which the basic motions of the body occur then. Initially, Ben's parents felt that Ben's development was much slower than others, but when Ben was tested for a viral infection at the age of 9, it was discovered that Ben's kidneys were slowly failing and he had multiple blockages in his heart. After this diagnosis, his treatment was started immediately, but unfortunately before the treatment could have any effect, he died of a heart attack. Now Now, why did this happen to Ben? Let us understand this very quickly in 2 minutes. Basically, mitochondria follow a 3-stage process called cellular respiration to produce ATPs. In its first stage, glucose produces 2 molecules of ATP in the cytoplasm through a 10-step glycolysis process whose end product is pyruvose. Now, after this pyruvate goes to mitochondria, where through the 8-step Krebs cycle, it again creates 200 ATPs, along with 2 byproducts, NADH2 and F8H2. Now, as you can see here, despite there being a total of 18 steps, it is made of only 4 molecules, whereas we know that one cell at one time produces a total of 38 molecules of ADP. So where are the remaining 34 ADP made? So well, for this, these byproducts NADH2 and F8H2 are done on these F1 particles of mitochondria. Here's 34 ADP are produced through the electron transport chain process. And in this process, basically, the high-energy electron series of electronic sectors present in NADH2 and FADH2 relist, that is, pass on, as if they are playing some passing the parcel game. And during this, their energy is quickly trapped in ATP. If mitochondria are defective, ATP production is limited to 2 forms, since both the Krebs cycle and ETC occur within the mitochondria. So what often happens in such cases? Well, to compensate for this ATP loss, the cells adopt Plan B, which is lactic acid fermentation QO, which our body usually uses only during vigorous exercises. That is, anaerobic form of energy generation. In this process, instead of the normal 10-step glycolysis of glucose, only 2 ATP forms will be formed in 3 steps. The byproduct of this process, lactic acid, does not break down further and starts accumulating in the muscles. And even during regular work, people suffering from mitochondrial genetic disorders feel as if they are exercising 24 hours a day because lactic acid starts accumulating in their muscles. So normally when you work out and your muscles get sore, you know that muscles get sore after exercise due to this lactic acid accumulation. The muscles are depleted of energy. Muscle feels fatigued. Now let me also ask you, if you exercise for 24 hours and lactic acid is constantly accumulating in your muscles, including the cardiac muscles of your heart, then tell me what will happen in this case. Obviously, heart failure and their death has happened with Ben also because lactic acid produces age plus ions, due to which the pH of the body becomes imbalanced, and now, as we know, different enzymes present in the body work at different pH levels. And it is exactly the same in the context of the body that we humans can neither work in extreme cold nor in extreme heat.
For best efficiency, optimum conditions are required, and same is the case with enzymes. And due to pH imbalance in the cells, they stop working. Its ultimate result is the cells becoming non-functional, which ultimately leads to organ failures. Upon discovering that mutated mitochondria caused the abnormalities, the scientist proposed replacing the defective mitochondria with healthy ones from another female via in vitro fertilization, IVF. But when this procedure was tried for the first time in 1996, the results were quite heartbreaking, because the brain of those born through this procedure was underdeveloped and defects started appearing in their main nuclear DNA. Whereas when the same procedure was tried on rats, the tests were completely successful. In fact, this whole process first started in the 1980s on rats, in which the entire cytoplasm, including cell organelles except the nucleus, were successfully transferred into the egg cells of other mice. In 1996, the procedure was authorized for human use following its success, but sadly, they saw its negative effects on humans, due to which FDN banned this process, and this concept remained just a theory. Until 2004, when the Human Fertilization and Embryology Authority, HFA, of UK showed its curiosity in this dimension, and with a minor change, they developed 2 new techniques through which the first 3-parent baby was born in 2016, who was immune to all diseases. Now the question arises that what did these UK scientists do? Well, the first technique among these is pro-nuclear transfer. Basically, this is a three-stage process of which the first stage is IVF. In this procedure, the doctor first takes one donor cell that contains healthy mitochondria, and then through IVF technique, one cell from both parent and donor is fertilized with the sperm of the male parent. During this fertilization, within about 12 to 18 hours, the pronuclear, which is made from the material of both the sperms, is extracted, because this is the time when the nuclear powers of both male and female are visible. With this, doctors can find out whether the genetic material passed from the parents is healthy, or if that pronuclear is healthy, then finally that pronuclear is transferred to a cell of the donor, and this gives birth to a healthy super baby. But there is a small problem in this. As you can see, the entire healthy embryo is discarded, which is unethical. So to tackle this, doctors invented another method, maternal spindle transfer. Well, this process is also similar to that. In this, the nucleus is removed before fertilization and transferred to a donor cell, and this entire process takes place in the most important phase of cell division, metaphase, because in this phase the chromosomes are aligned, due to which it becomes easy to segregate them and place defects in them. But the thing to think for a minute is that if the mother's mitochondria are defective, then why are the mitochondria of an unknown donor taken instead of directly taking the mitochondria of the father? So see, the answer to this is mitochondrial population, because whereas a sperm has 50 to 75 mitochondria, an egg cell has 100 million mitochondria. This is because a female cell is 400 times larger than a sperm, and that is why the donor should always be a female cell. Well, even if a stranger's cell is used, the actual rate here is still 70 to 80%. There's a 70-80% chance of a healthy super baby being born. But friends, every coin has two sides, as you know. I mean, even if the baby is now immune to the detected genetic disorder, there are still higher chances that when the fertilized egg is transferred to the mother's womb, there might be problems in embryonic development due to maternal age, environment factors, and health conditions. To prevent this from happening, scientists have also designed an artificial boom, in which all the nutrients required for the development of a baby will be present. Now, exactly how this artificial boom works is a revolutionary technology. It has already been explained completely in detail in this video. You should not miss it at all. It is a very interesting video. And friends, if you got to learn anything new by watching this video, then definitely give a like and subscribe to Sunfly Science channel, and press the description in the bell so that you do not miss any update of our upcoming videos. Until next time, stay curious and continue to learn and grow.